everyone, I am Elijah Dominic Soteo, and today we will explore liquid chromatography and its various separation techniques. This will be crucial for our studies in medical laboratory science, so today, let us find out what is liquid chromatography. It is also known as high performance liquid chromatography or HPLC. It is an analytical technique used to separate, identify, and quantify compounds in a mixture. It's widely used in pharmaceuticals, food safety, environmental monitoring, research, and in clinical diagnostics. Next, let us discuss the fundamental principles of liquid chromatography. HPLC separates compounds based on their interactions with two phases. First, we have the stationary phase. It is a solid or liquid-coated surface inside a column. And then the mobile phase, which is a liquid solvent that carries the sample through the column. Let us focus on the diagram here. It shows a basic overview of the HPLC process. The solvent used to separate components in a liquid sample for HPLC analysis is called the mobile phase. The mobile phase is delivered to a separation column, otherwise known as the stationary phase, and then to the detector at a stable flow rate controlled by the solvent delivery pump. A certain amount of the sample is injected into the column, and the compounds contained in the sample are separated. The compounds separated in the column are detected by a detector downstream of the column, and each compound is identified and quantified. That is the basic process of HPLC. Now let's have the 5 common separation techniques utilized in liquid chromatography. We have the reverse phase chromatography, normal phase chromatography, size exclusion chromatography, ion exchange chromatography, and affinity chromatography. Let us first discuss about the reverse phase chromatography. RPC separates compounds based on the hydrophobicity, wherein non-polar compounds elute first because they have weaker interactions with the polar stationary phase. This technique is commonly used for pharmaceuticals. Next, we have normal phase chromatography. In NPC, polar compounds elute first as they interact more strongly with the stationary phase. This method is useful for separating small polar molecules and purifying crude samples. Then we have size exclusion chromatography. SEC separates based on molecular size. Larger molecules elute first because they cannot enter the pores of the stationary phase. This technique is often used for separation of biomolecules like proteins, enzymes, nucleic acid, etc., and also industrial polymers. Next, we have ion exchange chromatography. IEC separates compounds based on charge. Analytes interact with charged groups in the stationary phase, which allows for effective purification and separation of biomolecules such as proteins, nucleic acids, peptides, and more. Lastly, we have affinity chromatography. This technique separates compounds depending on the specific affinity between the stationary phase and the analyte or the ligand. This method is particularly effective for isolating biomolecules like antibodies or enzymes. In summary, liquid chromatography or HPLC is a versatile technique that employs various separation methods tailored to specific applications in medical laboratory science and beyond. That is all for today po. Thank you very much for listening. If you want to check out the sources for my lecture, I provided the link here. Again, thank you very much and God bless.